Welcome back to another deep dive. We're really glad you could join us today.
Yeah, thrilled to be here.
Usually when we talk about a medical diagnosis, there's this expectation of like absolute precision.
Right, like it should be perfect.
Yeah, like engineering. If you break your arm, the x-ray shows that jagged white line on the screen, the doctor points to it and says, there it is. It's incredibly comforting. We like things to be visible, to be, well, perfectly categorized.
It feels definitive. I mean, the bone is broken or it isn't. You can see the mechanics of the problem right in front of you.
But then you step into the world of infectious diseases and you expect that same high-tech machinery. You expect flashing lights, DNA sequencers, computer readouts telling you exactly how to kill some dangerous superbug.
Which is almost never how it actually works on the ground.
Exactly. Instead, the reality of how we make some of the most critical life or death medical decisions comes down to something surprisingly low-tech. We are talking about a jelly-like agar plate seeded with bacteria, stamped with little paper discs of antibiotics, creating these mysterious clear rings.
Yeah, it looks like something you'd do in a high school biology class, honestly. Yet it is the absolute foundation of modern clinical microbiology.
It's wild to think about.
It is. That humble physical setup you're describing is on the front lines of one of the greatest threats to modern medicine, which is, of course, antibiotic resistance. Without those little paper discs and clear rings, doctors would essentially be fighting blind.
They'd just be guessing.
Pretty much, guessing which drugs might actually save a patient's life.
And that is exactly our mission for today's deep dive. We are looking at a stack of sources covering the Kirby-Bauer test, formerly known, I should say, as the Disc Diffusion Antibiotic Susceptibility Test.
Quite a mouthful.
Yeah, we'll stick to Kirby-Bauer. We're going to explore how this surprisingly simple, decades-old laboratory setup dictates exactly which drugs will work against an infection and why global health still relies so heavily on it today. Okay, let's unpack this. To really understand how this works, we have to look at the famous accident that inspired it all. Because, nobody just woke up and decided to put paper on jelly.
No, definitely not. We have to rewind to 1928 to a very, very messy lab bench at St. Mary's Hospital in London.
Classic Alexander Fleming.
Exactly. So Alexander Fleming returns from vacation. He had left a bunch of petri dishes out, completely seeded with Staphylococcus bacteria.
Gross.
Yeah, very bad lab practice by today's standards. Anyway, he finds that a stray mold, specifically a strain called Penicillium notatum, has drifted in through the window or up from another lab, and it contaminated one of his plaques.
The ultimate, fortunate mistake in medical history.
It really was. But what is crucial for our deep dive today isn't just the discovery of penicillin itself. It's the physical, visual phenomenon Fleming observed on that plate.
Right, what he actually saw.
Yeah, around that fuzzy blob of mold, there was a completely clear ring where the staph bacteria had simply melted away. They refused to grow.
And that's a ring we're talking about.
Precisely. That clear ring is what we call a zone of inhibition. The mold was naturally secreting this antibiotic substance that diffused outward into the gel, basically stopping the bacteria in its tracks.
Wow.
And that exact visual, a central source of a drug, creating a halo of bacterial death or inhibition, that is what the Kirby-Bauer test was later built to measure deliberately.
But reading through these sources, it is wild how long it took to go from Fleming's moldy plate to a unified medical test. It wasn't an overnight jump at all, right?
No, not even close.
The 1940s and 1950s are described here as a complete Wild West era for clinical antibiotics. So how did hospitals actually test these new miracle drugs before they had a standard rule book?
They essentially made it up as they went along. Yeah, as antibiotics transitioned from laboratory curiosities into mass production during and after World War II, hospitals realized they had a massive new problem.
Which was?
Well, a doctor would prescribe penicillin, but sometimes the patient didn't get better. The bacteria were already learning to resist it.
Oh, wow. Already?
Yes. So local hospital labs realized they needed to test the specific bacteria from their specific patients against these drugs. Dozens of hospitals just started running their own homemade versions of disc diffusion tests.
And homemade in a medical diagnostic context sounds like a recipe for absolute disaster.
It was complete clinical chaos. Every lab had a totally different method.
Like what? What were they changing?
Everything. They used completely different amounts of bacteria, what microbiologists call the inoculum size. One lab might, lightly swab a plate, and another might just drench it in a thick bacterial soup.
Oh, geez.
They poured their agar gel to different depths. The paper discs they used had wildly different concentrations of the drugs. Even the incubation times and temperatures varied depending on what kind of oven they had.
Which means the results were completely subjective. If I can use an analogy here, it's like baking.
Okay, let's hear it.
Well, if every baker uses a pinch of flour, a glug of milk, and just sets the oven to hot, nobody's cake comes out the same.
Exactly.
A doctor in Chicago might look at a homemade test and declare a bug resistant, while a doctor in New York, looking at the exact same organism with a slightly different homemade test, would call it susceptible.
And the danger there is that a diagnostic test's lack of credibility suddenly became a massive public health problem. The World Health Organization's Expert Committee on Antibiotics actually had to intervene.
The WHO got involved over this.
They had to. They flagged this lack of standardization as a severe global threat. I mean, you simply cannot track or treat resistant strains spreading across a country if no one is speaking the same diagnostic language.
Right, that makes total sense.
So the WHO desperately pushed the scientific community to create a single reproducible protocol.
Enter the heroes of our outline, William M. Kirby and Alfred W. Bauer.
Yes, along with their colleagues John C. Sherris and Marion Turk at the University of Washington in Seattle. In 1966, they published a landmark paper that truly changed everything. It was titled Antibiotic Susceptibility Testing by a Standardized Single-Disc Method.
I love looking at what they actually did here because they didn't invent the concept of the paper disc and they didn't invent the agar.
No, they didn't.
They just locked down the variables. They basically wrote the ultimate, strictly enforced recipe that every laboratory kitchen on Earth had to follow.
They standardized every single step to eliminate the chaos. First, they defined the bacterial density.
Okay.
Before you swab the plate, you have to mix the bacteria in liquid until the cloudiness matches something called a McFarland turbidity standard.
Ah, so you can't just guess the cloudiness.
Right. It ensures you are always testing the exact same number of bacterial cells. Second, they mandated a highly specific nutrient medium, which is called Mueller-Hinton agar.
Why that specific agar? Like, why not any other jelly?
Because it had a very consistent composition, and crucially, it lacked rogue substances like excessive calcium or magnesium.
What did those do?
They can chemically bind to certain drugs and completely interfere with the results.
Oh, I see.
Then, they locked in the precise incubation temperature and time. But the real genius of the Kirby-Bauer method wasn't just standardizing the setup. It was how they tied the physical size of those clear rings to hard mathematical data.
This is the part that fascinated me in the sources, because it's one thing to say, okay, the clear ring needs to be this big. It's another to prove why. How did they figure out exactly what a 15-millimeter ring meant versus, say, a 20-millimeter ring?
They did it through an incredibly tedious process of comparison. They took hundreds of bacterial strains and tested them using liquid broth methods.
Liquid tests.
Yeah, in liquid tests, you set up a row of test tubes. Each tube has a slightly lower concentration of the antibiotic. You find the exact tube, the exact lowest concentration of the drug that completely stops the bacteria from growing.
Okay.
That number is called the minimum inhibitory concentration, or MIC.
So they found the exact killing or stopping threshold for the drug in liquid.
Yes. And then they grew that same bacterial strain on an agar plate with a paper disc, incubated it, and measured the resulting clear ring with a ruler.
Ah, so they matched them up.
Exactly. By doing this thousands of times, plotting the millimeter zone sizes against the exact liquid MICs, they drew a statistical regression line. That allowed them to set the breakpoints.
Breakpoints.
Yeah, the specific millimeter cutoffs where a zone officially translates to resistant or susceptible. Their protocol was immediately endorsed by the WHO, and it remains the direct ancestor of the methods used worldwide today.
So now we have this strict, standardized recipe. But let's get into the mechanics. What is physically happening on that plate when you pop it in the incubator? How does a piece of paper reveal a bacteria's genetic secrets?
At its core, the test relies on the fundamental physics of diffusion.
Okay, picture time for the listener.
Right. Imagine dropping a heavy drop of dark blue ink onto the center of a wet paper towel. The ink immediately starts bleeding outward from the center, right?
Right, it's super dark in the middle, and it gets lighter and lighter the further out it bleeds.
That is exactly how the antibiotic disc works. You have a paper disc loaded with a highly concentrated fixed mass of a drug. The moment that disc touches the wet agar gel, the drug molecules start moving from that area of high concentration outward.
Into the gel.
Yes. Right next to the disc, the drug is incredibly concentrated. Farther away, it gets dilute, eventually approaching zero. Now remember, you've already evenly seeded the entire plate with the patient's bacteria, so every single spot on that agar has an equal chance to grow.
Except where the drug concentration is too strong for them to survive. So it's a race. The drug is bleeding outward, and the bacteria are trying to grow upward.
The bacteria will multiply everywhere until they hit a physical boundary where the local antibiotic concentration exceeds what they can tolerate.
That makes so much sense.
Yeah, that border of visible growth, the hard edge of the clear ring, marks the exact distance at which the drug's concentration has dropped to that specific bacterial strain's inhibitory threshold.
So a huge clear zone means the bacteria were stopped way out in the distant suburbs of the plate where the drug was super weak, meaning they're very susceptible.
Right.
But a tiny zone, or no zone at all, means they just marched right up to the disc, totally ignoring the drug.
Exactly.
Let's ground this with a concrete example from the sources. We have a lab strain of E. coli. You streak it on a plate, drop an ampicillin disc on it, what happens?
If it is a normal, susceptible wild-type E. coli, the drug bleeds out, the bacteria are highly sensitive to it, and you get a beautiful, wide, crisp, clear zone of inhibition.
But then you introduce a genetic twist. You introduce a plasmid. And for our listeners, a plasmid is basically a tiny floating ring of DNA that bacteria can pass back and forth to each other like trading cards.
That's a great way to describe it. So you take that same E. coli species, but this time it carries a plasmid containing a beta-lactamase gene.
Ooh, what does that do?
This gene is a set of instructions for the bacteria to manufacture an enzyme. That enzyme acts like a microscopic pair of molecular scissors. It physically hunts down the ampicillin molecules and cuts open their chemical structure,
Just chops them up.
specifically the beta-lactam ring, effectively destroying the drug.
And when you test that weaponized strain on a Kirby-Bauer plate,
It grows in a thick, dense lawn right up to the very edge of the ampicillin disc. It's a perfect visceral demonstration of horizontally transferred antibiotic resistance. You can literally see the resistance happening right in front of you.
it's amazing. But does this diffusion math ever fail? What happens if the bacteria just hide their resistance? Because the sources bring up MRSA, methicillin-resistant Staphylococcus aureus, which is a massive, terrifying hospital threat. But apparently, testing for MRSA with a Kirby-Bauer plate isn't as straightforward as just dropping a methicillin disc on it and looking for a ring.
No, it's actually one of the most fascinating diagnostic problems in microbiology. If you test MRSA directly with oxacillin or methicillin discs, the results can be incredibly deceptive.
Why is that?
The primary resistance mechanism in MRSA is driven by a gene called MECA. But MRSA exhibits a behavior we call heteroresistance.
Meaning that even though all the cells have the genetic code for resistance, only a tiny fraction of the population is actually expressing it at any given moment.
That is the fundamental problem. Most of the cells on the plate are acting like they are susceptible. Only a tiny rebellious subpopulation is expressing the MECA gene strongly enough to fight off the drug.
Wait, so if most of them die?
Exactly. Because most of the cells die, the clear zone on the plate can look deceptively large. A busy lab tech might mistakenly tell a doctor the bacteria is susceptible to standard drugs.
Which would lead to a catastrophic treatment failure in the patient.
Absolutely.
So how do you solve that? How do you force the bacteria to show their hand?
You use a pharmacological workaround. Microbiologists discovered that instead of testing with standard oxacillin, they should use a disc of a different drug called cefoxitin.
Cefoxitin.
Yeah, cefoxitin is in the same broader family of antibiotics, but it has a unique property. It acts as a powerful inducer of the MECA gene.
Oh, wow. It basically provokes the bacteria. It's like smoking a hornet's nest to see how many hornets are really in there.
That's exactly it. It forces all the MRSA cells to turn on their resistance gene at once.
That is brilliant.
It forces the entire resistant subpopulation to reveal itself reliably. When you use cefoxitin, that deceptively large zone shrinks down to a distinctly small resistant zone. The physics of diffusion didn't change at all, but we had to substitute the chemical to make the underlying hidden biology legible to the naked eye.
That is so incredibly clever. But wait, I have to jump in here and push back on something that seems, well, deeply counterintuitive. If I'm looking at one of these plates and there is a massive, totally clear halo around the disc, doesn't that definitively prove the antibiotic murdered every single bacterium in that specific zone? I mean, it looks totally barren.
It's a very common assumption, even among medical students. But no, it absolutely does not prove they are dead.
Really? How can they not be dead? There's nothing there.
There is no visible growth. That clear zone only proves growth inhibition. We call that a bacteriostatic effect.
Whoa, bacteriostatic.
Right. The drug stopped the bacteria from dividing and multiplying to the point where they could form visible colonies. It does not necessarily prove bacterial killing, which is a bactericidal effect.
So they could just be sitting there.
The bacteria in that clear zone might just be in a state of suspended animation. If you want to confirm that they are actually dead, you'd have to run a completely different liquid assay called the minimum bactericidal concentration, or MBC test.
That is a wildly important distinction, especially if you are treating a patient with a weak immune system who needs the drug to do the actual killing.
It is critical, yes.
Okay, so we're relying on these discs to give us accurate diffusion rings. But what if the drugs themselves go bad? Like if a shipment of antibiotic discs sat on a hot loading dock in the sun and lost half their potency, wouldn't they make a normal bug look like a superbug because the ring would be too small?
That is exactly why routine quality control is mandatory. Every single day before a lab can report patient results, they run the exact same Kirby-Bauer protocol using official, standardized reference bacteria from global culture collections. A famous example is a strain called E. coli ATCC 25922.
And what makes that specific strain so special?
It is a globally recognized, genetically identical clone. Every lab in the world has it. And because it never changes, we know exactly, down to the millimeter, how big the zone should be when it's exposed to a properly functioning antibiotic disc.
Oh, I see.
So if the lab tests that control strain, and the ring is, say, 3 millimeters smaller than it should be, they know the batch of discs has degraded from heat or moisture.
And they just pitch it?
They flag the bad batch and throw it out before it ever touches a patient's diagnostic sample.
So we have these perfect daily control plates ensuring the math is right and the drugs are fresh. But, the bacteria don't care about our math when they are inside a human body.
No, they certainly do not.
How does a clear ring on a piece of plastic translate to a living patient? Because deciding how big a ring needs to be to count as a win for the drug seems to be where the hard science meets some very messy human judgment.
It is the philosophical core of susceptibility testing. Originally, decades ago, people treated the word susceptible as a fixed biological property, like a law of nature.
Right.
Either the bug was intrinsically weak to the drug or it wasn't.
But the sources make it clear that's not how modern medicine views it at all.
Not at all. The modern reality is that the breakpoints, those millimeter cutoffs that divide the categories of susceptible, intermediate, and resistant, they are moving targets.
Moving target.
Yes. They are not laws of nature. They are set and constantly debated by massive expert committees. In the United States, this is handled by the CLSI, the Clinical and Laboratory Standards Institute. In Europe, it's EUCAST, the European Committee on Antimicrobial Susceptibility Testing.
Here's where it gets really interesting. So susceptibility isn't just a biological fact about the bacteria.
Right.
It's a probabilistic, revisable clinical judgment about a standard dose at a typical infection site. It's a moving statistical target dressed up as a hard laboratory fact.
That is a beautiful synthesis. These committees look at the raw physical zone data, but then they weigh it against incredibly complex human factors.
Like what kind of factors?
Like, how is the drug metabolized in the human liver? What concentration of the drug actually reaches the blood or the lung tissue safely without poisoning the patient? How fast are new resistance genes spreading in the global population? All of this influences the millimeter cutoff.
And the sources note that CLSI and UCAST actually disagree sometimes.
They do.
You could have a 15 millimeter zone of inhibition on the exact same plate on the exact same day, and the European Committee might officially classify it as susceptible, while the U.S. Committee stamps it as resistant, based entirely on how they interpret the clinical pharmacology.
It highlights just how much clinical human judgment is baked into this supposedly objective test. They are constantly holding meetings and revising these reference tables as new population data comes in.
So if the definitions are this fuzzy and the clinical judgments vary across the Atlantic, what are the limits of this test? It's a 60-year-old standardized recipe. When do doctors look at a Kirby-Bauer plate and say, this isn't enough, I need something else?
The biggest limitation is that Kirby-Bauer is qualitative, or at best, semi-quantitative. It puts the bacteria into a bucket, susceptible, intermediate, or resistant. It does not tell the doctor the precise MIC number.
The exact lowest concentration that stops growth, like we talked about earlier.
Right.
But why does a doctor need that exact number? If it says susceptible, isn't that good enough?
For a routine skin infection, yeah, it usually is. But for severe, life-threatening scenarios like bacterial meningitis or a bloodstream infection, doctors need the exact MIC to calculate highly aggressive, perfectly safe dosing.
Oh, because the margins are so tight.
Exactly. Knowing exactly how far above or below a break point that organism sits can dictate whether the patient survives the night.
So how do they get that exact number without doing those tedious rows of liquid test tubes?
They turn to other methods. One brilliant innovation from the 1980s is called the E-test.
Ah, I saw pictures of this in the sources. It looks like a little plastic ruler. How does it work?
It's a plastic gradient strip that you lay on the agar, just like a disc.
Hmm.
But instead of one fixed dose of the drug, the plastic strip is impregnated with a continuous decreasing concentration of the antibiotic along its entire length.
So one end has a massive dose and the other end has almost none.
Precisely.
So instead of a perfect circle, what kind of clear zone does that make?
It creates this beautiful teardrop-shaped zone of inhibition. The teardrop gets narrower as the drug concentration drops along the strip. The genius is that the strip has a numbered scale printed right on top of it.
Oh, so you just read it.
Yeah, you just look at where the edge of the bacterial growth intersects the plastic strip, and you literally read the exact MIC number off the ruler. It seamlessly combines the physical simplicity of the Kirby-Bauer plate with the quantitative precision of liquid broth.
That's amazing. But the sources also mention that Kirby-Bauer completely fails on certain types of bugs. Like, you can't use it for slow growers like tuberculosis. Why is that? What's stopping a slow bug from making a clear ring?
It comes down to the biology of the bacteria and the chemistry of the agar. Mycobacterium tuberculosis has an incredibly thick, waxy cell wall. It divides very, very slowly. A standard E. coli might divide every 20 minutes, forming a visible lawn in 24 hours. TB can take weeks to form visible growth.
Oh, I see. And by the time the TB finally grows enough to be visible.
The antibiotic in the paper disc has completely degraded. It broke down at room temperature weeks ago. So the ring you get is completely meaningless.
Ah, that makes sense.
For TB, strict anaerobes that die in oxygen, or highly fastidious bugs that require complex nutrients instead of standard Mueller-Hinton agar, you have to use specialized molecular genetic testing.
Okay, let's clear up a few major misconceptions that the sources highlight, because this completely blew my mind.
Go for it.
I always assumed that if I looked at a plate and saw a massive clear zone, that automatically meant it was a stronger or better drug than one with a small zone.
What's fascinating here is how deeply ingrained that misunderstanding is. You simply cannot compare the zone sizes of different antibiotics to each other.
So if a drug is just a smaller molecule physically, it diffuses faster and further through the jelly and makes a bigger ring. Comparing a 20-millimeter ampicillin ring to a 15-millimeter tetracycline ring is totally meaningless unless you look at their specific individual break point tables, right?
It's an apples to oranges comparison. The raw millimeter size only matters when compared against that specific drug's own established standard.
Okay, misconception number 2. If a bug is labeled resistant on the test, it means the drug is biochemically incapable of hurting it under any circumstances, like it has a force field.
If we connect this back to human anatomy, we realize that resistance is relative to where the drug is going. A bacteria might be labeled resistant based on the drug concentrations we can safely achieve in the human bloodstream.
Right.
But if that same drug is known to concentrate heavily in the bladder when the body filters it out, it might easily cure a urinary tract infection caused by that exact same bacteria.
Wow.
The categorization is tied to standard doses and typical infection sites, not absolute biochemical invincibility.
Which is why the CLSI actually publishes some breakpoints that are specifically calibrated just for urine.
Exactly.
And finally, because this test is a standardized recipe from 1966, some people assume it's an outdated relic that's been totally replaced by fancy computers.
While automated susceptibility systems, like the high-throughput VATEC machines, are common in massive Western hospitals, Kirby-Bauer is far from a relic.
Yeah, the sources were very clear about that.
It remains a global standard because it is profoundly democratic, it is incredibly cheap, it is easy to standardize, and it requires almost no specialized gear, an incubator, and a ruler. In low-resource settings around the world, this test is the primary firewall against untreatable infections.
Which brings us perfectly to the global picture. Why does this ruler and agar test matter to you listening to this right now?
Well, every time you or someone you love is prescribed an antibiotic based on a culture rather than just a doctor's educated guess, it's thanks to this exact testing logic.
Yes.
But it goes beyond the individual patient.
Yeah, it scales up massively. Hospitals take all these individual Kirby-Bauer results from thousands of patients and aggregate them into reports called antibiograms.
Antibiogram. Right. When you show up at the ER with a severe mystery infection, the doctor can't wait 48 hours for your specific plate to grow. They look at the hospital's antibiogram to see what antibiotics are generally working against local bacterial populations that specific week, and they start you on empiric therapy based on that data.
It's essentially the local weather report for superbugs.
That's a great way to put it.
And on a macro level, it is the primary tool the WHO uses to track the overuse-driven resistance crisis. We are talking about tracking MRSA, or CRE, carbapenem-resistant Enterobacteriaceae, which are terrifying gut bacteria that have learned to resist our absolute last resort drugs.
Truly scary stuff.
Without this humble paper disc test, our global antibiotic stewardship would be pure guesswork.
But the system only works because we had standardized it and we have those expert committees constantly updating the rules. It requires immense ongoing global coordination.
Which leaves us with this final thought for you to mull over. If our definition of a susceptible infection keeps changing based on human drug delivery, and breakpoints rely on expert committees arguing over data in conference rooms, and resistance is a constantly moving target, well, what happens when the bacteria start evolving faster than our human committees can convene to print new reference tables?
Yeah, when does the messy biological reality outpace the standardized recipe? It's a sobering thought. The bacteria certainly don't wait for the CLSI to publish an annual update.
They absolutely do not. That's all we have for today's deep dive into the Kirby-Bauer test. Keep questioning the obvious, and we'll see you next time.